I'd like to introduce Dr. Francesca Schiavone, who will be speaking on operational aspects of adaptive platform trials. Dr. Schiavone is based at the Medical Research Council Clinical Trials at University College London, overseeing the planning and conduct of international clinical trials, particularly in resource-limited settings. Since joining the MRC CTU in 2012, she has worked on several adaptive platform trials in oncology and infectious diseases. She is currently involved with the setup and conduct of 2 large international trials utilizing a practical design. Dr. Schiavone is also involved in the MSc in clinical trials run by the Institute of Clinical Trials and Methodology. She is a member of the MRC-NIHR Trials Methodology Research Partnership Working Group on Data Quality and Monitoring, and is a protocol editor at Trials. Please welcome Dr. Schiavone.
Thank you very much, everyone.
Very happy to be here today. I'm sorry I can't be there with you in person. I was trying to kind of create some sort of emotional bonding over the fact that I had a very troubled start to the week, and I wanted to kind of milk for sympathy. Thank you for the opportunity, and as I said, thank you to the speakers earlier today. It's been a really interesting conversation and discussion around adaptive platform trials. I'm going to share my experience here on some of the trials I've worked on, obviously adaptive platform in nature. I'm going to, as much as possible, to also really share the legacy that the MRC Clinical Trials Unit has on the topic. And I would like to specifically focus on challenges and solutions from a trial conduct perspective. And I like really to open the floor to questions about opportunities that lie ahead of us. So, as I don't like to sort of make assumptions about knowing what the MRC Clinical Trials Unit is, I put here just a very simple tagline, which is the tagline of our trials unit. It's in London. What we do is that we specialize in the design, analysis, and conduct of clinical trials, specifically with an interest in methodological innovation with adaptive designs. We are part of UCL, University College London, and we are the founding a unit part of the Institute of Clinical Trials and Methodology. So just to give a little bit of a context about who we are an academic unit, and although there are strong industry partnerships, ultimately we come from an academic background, as you would expect from a CTU. And it's a very small picture, and of course it doesn't represent the over 200 employees that are part of the MRC Clinical Trials Unit, but you'll see here in a very small faces, but a group of mostly statisticians, but also clinicians and trialists in general at the last MRC Clinical Trials Unit MAMS symposium, and you will see here the I would say the founding father of MAMS design with binary outcomes, Max Palmer, you see here, and I'll refer to him as well as Sarah Walker, you see here, when talking about the practical design as well. So some faces, but there's many more of us around. So really what I'm going to be talking about today. So first of all, I'd like to sort of start with a very brief overview of some lessons that I've learned specifically in MAMS design trials. And I'm going to bring the experience that I have hopefully sort of matured over time with Stampede, which is a advanced prostate cancer trial for the treatment of prostate cancer, now closed and has recruited over 12,000 patients over the space of actually like 15 to 20 years. So quite a long longevity, and also biomarker stratified trial focus 4, which actually uses a multi-comparisons design, and again in a late oncology settings. But really what I would like to bring together is the experience that I'm really is quite new at the moment, and this is why it would be really good to have discussions with you all, which is about new questions being addressed with new designs. And specifically I'm going to be looking at the challenges posed by antimicrobial resistance, as well as looking at neglected tropical diseases like snake bites, but in the context of a practical design trial, which is a new innovative design. So I don't like dichotomy, and I don't think it's a helpful way of thinking about this. But one of the first thing I was sort of going with, and has been eminently explained with the first talk of today, really is kind of the binary distinction between a traditional two-arm design, whether it's blinded or unblinded. Ultimately, it's non-important at this point. And then sort of the other side of the coin that is considered to be innovative trial designs. And that's just kind of a very, very simple, very crude sort of division of how randomized controlled trials work. But we know ultimately that the gold standard for evidence-based medicine is in randomized controlled trials, and there is an agreement. Now, something that I think is a little bit counterintuitive given the audience and the forum today is that I would like to sort of spare a word towards like caution. And the thing that I would like to say is that we don't always have to use an adaptive platform trial design. I think there are numerous good, valid reasons for wanting to opt for a traditional design, a simpler sort of two-arm comparisons without the adaptive platform element to it. And it's important not to be, I think, to some extent, lured into the sexiness of these designs, because obviously there are complexities, and it's not to say that they can really answer all the questions there are. They're very good at answering questions efficiently, and there's been numerous examples today already, but really sort of a word of caution that we don't always have to sort of migrate towards an adaptive platform trial design just because it's fancy. And of course, we can look at this in different ways. And when very broadly I talk about innovative trial designs, really, I can probably put in a lot of different statistical designs. Opinions might differ, of course, but traditionally we don't think about umbrellas and baskets as having potentially an element as being adaptive and being a platform. But really, there are efficiencies there. We can go with a more traditional and perhaps a bit more holistic definition of a platform protocol where you change stuff and you have a formal approach with interim analysis with a data monitoring committee. You can look at the MAMS sort of arena, both in its traditional fashion, but also in the MAMS selection design, as well as the MAMS Rocky design, which is a duration design. And something that I would like to touch on within a little bit more detail today is not so much about the MAMS area, but really looking at Practical as a new trial design that I hope will evoke some interest in the audience. And this is a very simple sort of distinction that has been repeated quite a few times already today. And just sort of drawing some comparisons between, for instance, and some differences between basket umbrella trials and a traditional platform trial where you have a shared control arm, the potential of dropping arms and adding new interventions and hurdles that really are pre-established and pre-planned via interim analysis. And I think we're all in agreement with sort of this sort of shape of a platform trial. And like many of you, I have found myself trying to kind of really wanting to boil it down to what are the key characteristics of adaptive platforms. One that I really, really like about Palman and colleagues has been planning to be flexible, because it really, really echoes of the operational complexities that come with adaptive platform trials. But one that I also really like is the one that's been sort of pushed forward by Max Palmer, our director, which really is looking at testing multiple primary questions within one protocol as a possible definition of what we mean about adaptive platforms. But these are just some of the definitions, and many of you have contributed to many others. And of course, there are some, again, components that will be coming back over and over again when we talk about adaptive platforms: the addition of a new comparison, the early stopping due to interim analysis for lack of benefit or overwhelming benefit, the operational complexities that come with it. So one very, very simple message that I think has been so nicely put forward by everyone today is that, of course, there's been a huge increase in the use of MAMS design and adaptive platform in general. And this is too small to read, but it's really mapping out 60 MAMS trials over the course of about 20 years. And you can see the uptake and the increase in popularity, if you like. And a different way of looking at it is in this figure that looks at sort of trials that have been where the sort of adaptive platform trials, specifically MAMS in nature, are being led from. And this is the picture at the top, picture A, and also where is being conducted pre and post-COVID as well. So you can see there is a global reach, and that's undoubted, without a doubt. But obviously we got far more sort of to cover globally. And of course, COVID research and the pandemic, unfortunately at the same time, has really thrusted through the use of MAMS. There are some brilliant examples that have been mentioned today, some of which are coming from the UK. Think about the recovery trial. Started off as a 4-arm trial with other arms added later. Astonishingly recruited 12,000 participants over a space of 100 days. It was a huge machinery, and it was a perfect embodiment, I think, of how public health policy really linked in with clinical trials, particularly of this nature, and influenced practice hugely, of course, during the pandemic. But there are other examples with the solidarity trials advocated by the WHO, run in over 52 or 53 countries. So a real global endeavor to address important research questions during the COVID-19 pandemic. And I think that's a very important reflection, and really we take from today. And as I said, I will only briefly touch on some of the work that we have published in terms of running late phase oncology trials. Again, these are the STAMPEDE trial in advanced prostate cancer and the FOCUS 4 trial in colorectal cancer, in fact. And we have quite been a little bit inventive with the titles of these papers and trials. That's really the work of my previous boss Matt Sides. We looked at the operational complexities of adding a new research component to an ongoing platform, specifically with examples from STAMPEDE and FOCUS 4, but also we have a focus on the data management and database development, something that is perhaps not talked about enough, but it's key for the efficiency of the platform and is demonstrated by STAMPEDE and FOCUS 4, I think, extremely well. We also have put some work together. This is very UK-centric in the sense that brings together the experience from UK Clinical Trials Unit in late phase platform sort of running. It's not specific to oncology, but there is a lot of experience coming from oncology in this arena. And again, we have distilled, if you like, 17 sort of key lessons about the conduct of this platform, adaptive platform trials. They are run globally, but really are led by the UK. And I think something that is not really talked about enough, and I would like to spend a few words on, is the emotional toll that running these trials really has on people. I'm not doing this just to create empathy, but really because there is a cost associated with the resourcing that is necessary for the effective running of an adaptive platform trials. And Liz Morell and colleagues at Oxford Uni have done this piece of work together with us at the CTU. I was one of the people that they interviewed, and it was a good opportunity to share really some of the how some of the complexities really were managed from an individual perspective as well as a team perspective. So I think there's some really good sort of food for thought in there, and it's quite unusual to sort of look at the emotional side of running these trials. And I will sort of encourage you to look back at the papers, but really we distill the knowledge around sort of protocol development, database setup, ethics and regulatory, contracts and drug supply as unique sort of components that really differ in adaptive platform trials compared to a traditional design. A lot of this will be echoed and repeated sort of throughout this presentation when we talk a little bit more about practical design and what it is. And most of which perhaps won't surprise you, but I think what I'm hoping to convey is the complexities of managing these different components, and again, sort of bringing back some and echoing some of the words from previous speakers about how really it's a multidisciplinary effort that is in place to run these trials, where there are experts in different domains. There are clinicians, statisticians, operational leads, and they all come together to execute these type of trials. But as I've been mentioning, I have been focusing more recently work around new research questions that are addressed actually with new statistical designs. One disclaimer I should have made at the beginning, I'm not a statistician or a clinician, and my work here is also presented through really effectively the work that's been done by brilliant statisticians like Professor Sarah Walker at the MRC CTU. So what is this thing about new designs? And I think I'll start with one of the assumptions that I think all of us make, and I certainly have and made in the past when talking about clinical trials. And the idea is that what we want to aim or what we want to achieve is to have one regimen that can rule them all. Essentially, you have a standard of care, a control that is aiming in a perfect efficacy trial scenario to maximize a good outcome and minimizing the bad outcome. So you think about toxicities as best as we can. It's the balance between these two that really we always play on. And then it sort of feeds into sort of what guidelines sort of say, and is generally aimed to fit most patients as best as one can. Obviously, we can do this iteratively in a design like a MAMS design, but other just platform designs where we want to improve this standard of care and optimize the regimen. We've done it in TB, we've done it in cancer, we've done it in HIV. And obviously, the innovation that adaptive platform trials bring is that you compare multiple new interventions at the same time. You have a shared control arm, which typically can be the standard of care. You have the efficiency there potentially, which is obviously very well documented, of very fast recruitment in some scenarios, but also the efficiency of the shared control arm, which sort of brings a bit of economy of participants that need to be randomized into a trial, and that shared contemporaneous control arm that really, really helps. But so really normally what we're doing is to look at sort of clinical guidance, clinical practice, clinical guidelines in order to choose our standard of care. And that's a legitimate approach and one that I wouldn't fault anyone for doing. But what happens if there isn't actually a standard of care? And that's something that I had to sort of face, I guess, early on in my career, is something that sort of baffled me a little bit to begin with, and probably I'm showing my inexperience here. And a really compelling example comes from antimicrobial resistance. We know it's incredibly well documented and published that there is a rising antimicrobial resistance, AMR. This is leading to an increasing number of multi-drug resistance bacterial infections, particularly in the gram-negative arena. We tend to use stronger and stronger antibiotics, which are used empirically, and that's not a good thing when we're working in a context of AMR. And of course, what we do see is as well, specifically an increase in carbapenem resistance infections, particularly in low-income settings and resource-constrained settings, but particularly in pediatric and neonatal populations, as they tend to be hit by the fatality quite a lot harder. And this is why Sarah Walker and colleagues have developed the personalized randomized control trial design, which is a new paradigm in randomized control trials. And I encourage you to refer back to this paper that explains really how this case is made so compellingly when it comes to carbapenem resistance infections. So the thing about antibiotics and AMR is that they're complex in themselves because there is a large number of antimicrobials, antibiotics. There's different durations that people play with, as I said, sometimes empirically. And in fact, there is a very, very weak evidence base. And I've been astonished to learn, and again, showing my inexperience here, that there are some products out there in antibiotics that are licensed really with data from one participant only. So it's really quite an immature, I guess, arena sometimes to operate in evidence base. But there are some very strong opinions, particularly among pediatricians and neonatologists, traditionally because babies and children are not really being looked at when it comes to antibiotics as much as is true for many other therapeutics. But also really is that any one standard of care is not really possible for many patients. And this is a real challenge here because antimicrobial resistance is generally mosaic, cross-class. There are resistant genes that sort of very mobile. There's also the recognition that many antibiotics have some very significant contraindications and side effects. And one of the challenges that comes in public health is also that typically, you don't really isolate a pathogen that is causing that infection very easily, and really the positivity rate it tends to be quite low. You also have some very serious conditions, particularly when you're thinking about non-complicated UTI, multi-drug resistance infections, where potentially really you have very substandard standard of care that you're working with. So that's all of this is really quite challenging, and that's inherently brought by the disease area, which is obviously serious bacterial infections. So in the practical design, there isn't a standard of care. Each patient is randomized between a subset of regimens, and these subset of regimens differ from patient to patient. So in a traditional platform design, there's still one standard of care, which every patient can potentially be randomized to. But really what's happening here is that you're not doing that on a one-to-one basis in a sort of pairwise comparison. But in practice, what you're doing, and I'm going to push this argument a little bit further, in a context of neonatal sepsis, which is a severe gram-negative infections in babies, what we're turning the questions to be is not if A is better than B, but really to identify one of the best out of multiple options for a particular patient. And more importantly, it's about avoiding the worst option, taking into account the toxicity, resistant patterns, availability of antibiotics, which is very important, particularly in resource-constrained settings, as well as cost. And there's a huge piece of work around health economics in this type of design that really is about to start. And I think it's, unfortunately, I'm not an expert, but I think it brings some important questions when it comes to public health. So really what the practical design does is that utilizes a network meta-analysis analogy. So you're looking at combining evidence for multiple treatments, which you are exploring via both direct and indirect evidence in a network in a statistically principled way. And really what you're trying to answer here, once again, is what is the ranking of the available treatments for the particular characteristic of that baby or that particular clinical subgroup that one has in mind. And I'll bring a couple examples as well from a different disease area with snake bites. I don't want to indulge too much on the design, but I like to say that the experience that I like to bring here is over the NEOSEP1 trial, which is the first trial that is being run using a practical design and is being implemented, is designed by the MRC Clinical Trials Unit. It's funded and sponsored by GARP, which is a global antibiotic research and development partnership, and as well input and clinical leadership from St. George's University of London. And one of the things that you'll hear is that we have run this trial in two parts, with the first part being completed and being a confirmatory PK on 60 babies across 3 sites in Africa. And we're looking at the combination of 2 old antibiotics. So we're not looking at novel regimens, we're looking at old regimens. So we're looking at fosfomycin and flumoxif, both using combination with each other as well as amikacin. And that's, as I said, that part 1 of the study has been closed and as reported just a few months ago. And the paper is in the making. So what we spent a significant amount of time, and this is something that again goes back to talking about resourcing in these trials, is the setup of part 2, which really started with a site feasibility assessment across both Africa, Sub-Saharan Africa, as well as the Asia Pacific areas, which has taken over 2 years. And I'm not too ashamed to say, it's just the complexity of it all that is really coming through. And what we've done is to look at identifying locally relevant both first and second line treatments for neonatal sepsis in babies that who are less than a month of age. And we're looking at a pool of treatments from WHO recommended antibiotic regimens, broad spectrum antibiotics, as well as the experimental regimen of interest that I mentioned earlier, fosfomycin and flumoxif. And a second part of the setup of this trial has really been within each site to identify what type of participants we're dealing with. Are there any resistant patterns that are known and established at the site, any facility-specific risks to resistance? What type of babies do they look at, and do they normally admit? Are they preterm or term? Are they inborn, outborn? They present different characteristics, and therefore they have a different type of randomization that really is implemented there. We also look at the very broad division of whether it's early onset or late onset, which really would sort of determine whether it's a sort of a vertical transmission or something that's really been coming from potentially from the hospital settings. So this is again the design and the schema of NIHR Part 2, which, as I said, is the practical component of the trial. Sites have to choose among up to 8 treatments from this list of pre-selected treatments that reflects broadly, obviously, the WHO guidelines. What they do, they will screen the patients, they will go to first-line randomization very early on, because we're looking at potentially an emergency setting with a very serious infections in these babies. We're looking at daily treatments daily treatment and observations while in hospital. And then really what you can happen is that typically sort of you continue to follow them up as we do up to day 28 and 90 for mortality outcomes. So day 28 is the primary outcome measure in this trial. So we're not looking at a microbiological outcome measure at all, because we want this to be a public health trial relevant to sites in Sub-Saharan Africa and Asia Pacific. I'm going to very quickly demonstrate really what it means in practice once we have a site randomizing. And this is the list of the possible treatments that a site can choose from. They have a baby in front of them that they deem to have early onset sepsis, and they decided that for this particular type of baby with these clinical characteristics, and this is a simplification mostly, they want to randomize among one of 4 treatments presented here, so the ones that are not crossed out. But for a different type of baby, which has, for example, late onset sepsis, we're looking at a much broader spectrum, removing sort of treatments like Ampangent or meropenem, but opening the randomization to a number of other regimens for late onset. So as I was saying earlier, what typically happens is that you follow up the baby, you check if it's improving or not getting worse. I think the concept of cure is really quite a little bit dodgy when it comes to neonatal sepsis. But let's say they don't get worse or they're getting better, and then you complete follow up. But really, what we have observed in the NIHR study, which is an observational study run globally with over 3,000 babies enrolled to it, that 25% of babies really declines and deteriorates typically quite quickly as well. And therefore, they need to access a second line randomization to escalate the treatment potentially. And this is where the NIHR-1 trial not only has a practical component, as just explained, but also a smart design component, where what we're looking to do is to have a list of possible randomization options for second line following deterioration, which obviously excludes what has been given in first line. And again, it becomes sort of a question about second line treatment and escalation. And the patterns of behavior in the trial, including the switching behavior, it's something that will be looked at very, very carefully by the statisticians. As I mentioned, we completed part 1 successfully, and we are in the process of potentially recruiting up to 3,000 babies across 16 sites, and expected to open in December this year. So we are really in the thick of it at the moment. One of the things I wanted to highlight is that to get to the point of defining a list of suitable randomization options for specific, site-specific clinical subgroup, we had to do a work which, as I said, has taken over 2 years. And this is just an example. It's very small, I realize this, but it's the list of options that have been selected and signed off by the principal investigator at Baragwana Hospital, one of the largest hospitals in the world in South Africa, Johannesburg. This is a 6 pages long document that really is the result of a very, very long conversation to really bottom out what we're going to do both in first line and second line for different babies at the sites once the trial is up and running. And this will determine how the randomization system will be set up, and at the moment is being tested, which is huge fun. So just to touch on the lessons learned, as I said, there is a lot that we already had learned from MAMS trials, and something that has been already so well discussed earlier today is over the protocol structure and how key that is to successful implementation. We need to engage with regulators and policymakers from the outset, which is something that we have done in USEP 1, particularly because this is a new trial, particularly because potentially this trial could contribute to the licensing or extension of licensing of some therapeutics, but also because this is a public health trial and it's important that the results are will contribute to WHO guidelines, and therefore that conversation really needs to start early on. We have also decided to obviously uncouple the PK confirmatory element from the randomized trial, but do that efficiently under the same protocol. So the implementation of practical really is just an amendment essentially into across all sites. And there's also the component of the possibility of adapting this trial as new treatment regimens sort of come for our attention. So there is a constant horizon scanning that really happens across all antibiotics to see what else can come in. And we have developed, of course, the flexibility of having country-specific appendices for the specific antibiotic regimen list. And again, this is a very small snapshot of the master copy. And again, this is where earlier we were saying about having the structure of master and sort of domain-specific protocols. They can be called in different ways. The functionality is slightly different, but version control remains one of the constants that we have to chase because, of course, implementation has to be as standard as consistent as possible across all sites. It's a complex design, and with that comes a huge educational package for sites, and not only for sites. We have a podcast that has been developed with investigators from the Part 1 trial, but also from the observational study that I mentioned earlier that created some of the data that this trial is underpinned on, NIHR-OBS. We have had some very interesting workshops with investigators and continue to have them. We call them antibiotics workshops, but really they are about possible randomization scenarios and using the randomization system in testing, of course, to be able to understand the concepts behind it, because it's new and because in general, randomized controlled trials are complicated anyway. And of course, this one-to-one, very personalized contact with sites to understand local practice, to see what kind of babies they have in front of them and what treatments they have available. We're working globally. We're working with international sites and partners. And of course, there's going to be challenges in contract negotiation or communication, which is why we advise generally to start small before we go big. There is the unpredictability of industry partners, something that I haven't mentioned so explicitly. These are academic trials, but they also be running with the support and at times partnership of industry or pharmaceutical companies. For instance, in USEP, that's Shinogi and Infectopharm, who supply flumoxif and fosfomycin. So there is something that we have to deal with. It's not something that we had specifically to do for USEP 1, but we're very, very wary of the fact that obviously there could be a change in scope by the industry partner that could change quite significantly the direction of a trial. And of course, if you're working with multiple partners, there is always the risk of duplication. So it's very important that you nail down from the very beginning a very clear division of responsibilities. And this is something that we've done with our partners at GRP and St. George's over the course of 2 to 3 years. And it's not as easy as it seems to be, particularly for new collaborations. And I think something that I want to say that, of course, is my own personal agenda, and I'm not going to hide it for a moment, is the fact that we are running here a public health trial. We're not running a regulatory trial. And of course, the expectations and demands on how it's run should be proportionate to that. Of course, there is complex design and randomization, as I said, and it's a continuous learning with sites. We need to have fundamentally a aligned strategic outlook. It's a long-term commitment to a public health goal, something that is set by the WHO. We're looking at developing, yes, of course, an adaptive platform, but we're here to create collaboration and networking that can help the future of other trials. We want to be pragmatic. We want to sort of run a public health trial. But of course, adaptive trials are challenging, and there is much more time dedicated to data management, clinical operations, clinicians, statisticians, and of course, the efficiency of these trials, particularly when we're looking at designs like MAMS, is well documented. We know they're efficient, but there are there is a requirement for some significant increase in funding for stats, for operations, for clinical expertise, particularly at the setup stage, which is not something that traditionally we have done. And I think it's something that it's important to remember when we're talking, for example, about how to interface ourselves with funders and what that means. And in summary, when it comes to NeoSEP 1, I have learned that at the beginning, I thought, of course, the database setup is going to be difficult. Of course, the treatment selection is going to be difficult. And of course, the randomization system is going to be the first one of its kind. It's going to be hard. But really, there is a different layer to the operations and the clinical operations around the trial conduct, which is to be sure to have a risk proportionate management approach, to always future-proof the conduct, which is very easy to say, but less to do. And of course, the fact that you're really moving into a sphere where you have complex project management. And I would say, in some cases, we're talking about program management, which is slightly different. And I'll take a few minutes to just talk about a different kind of platform. And this is different because there is an adaptive design component to it, but is a little bit different because it's not the only component. This is SNP Africa. It's a Pan-African collaboration funded by EDCTP3. For those of you who are not familiar with it, it's a global health sort of funding initiative supported, well, essentially by the European Union to support infrastructure, research infrastructure in Africa. We're looking here at 3 components of the platform: a clinical and microbiological surveillance study, so not an RCT, a PK study, and then the complex adaptive drug regimen trial, which is in fact NeoSEP. So NeoSEP is actually embedded in this platform. And there are some cross-cutting themes like training and capacity building, stakeholder engagement, and integration. This work is led by Penta, which is a no-profit research organization based in Italy, with the input and partnership, of course, of the MRC Clinical Trials Unit, St. George's, GARPI in Switzerland, as well as our African partners. So what would he look in practice? And that's something that we're really coming to grasp with, because it's just really been funded about 2 years ago, and there's still a lot of lessons we're learning. So imagine, as I said, we have the work package, the component of the domain that is looking at confirmatory studies on the dose of an antibiotic, which could be an old antibiotic, but just been experimented in a neonatal settings, which is exactly the scope of SNP Africa, exactly as NeoSEP. So the dose for the antibiotic is tested and confirmed within the platform, and then is then feeding into the neonatal trial with NeoSEP and added as a potential agent of interest among the randomization options. So there is that sort of feeding mechanism of the platform itself that, although it's not entirely adaptive statistically in nature, a component is, and you see how different parts interact with each other to really address the question about what works best for babies with severe sepsis. And it's an exciting challenge, particularly because the number of partners and the fact that it's the first of its kind to be run. So it's an exciting project to be involved. But then we talked about sort of new questions and new designs, but practical. But now I'm kind of sort of changing a little bit school and going, there's different questions that can be addressed, of course, by the same design with the practical design. And I never thought in my life I was going to be working on a trial on snake bites, but here I am, and I'm delighted. I'll start by saying that there are up to 250 snake bites or snakes that are of medical importance. And we know, and some of us have some very well-established phobias with snakes. There's huge variability in size, in shape, in behavior. They're highly adapted to their environment. But really what I discovered, which I did not know, is that it's a neglected tropical disease, and it has the highest disability-adjusted life years among all the tropical diseases that have been investigated. The treatment for snake bites, it will not surprise you, is the administration of antivenom, which has been the case for about 100 years, but there's huge challenges when it comes to their efficacy, their quality, and also their safety. So much so there are a number of antivenoms on the market being used at the moment on real patients, not clinical trial patients, which have no data whatsoever to support a risk-benefit assessment. It's a very strange landscape to be in. And this is why also in 2019, the WHO has taken a look a little bit about what was around and came to put together a strategy for prevention and control of snake bite and venoming. What they did is that they looked at about how poor the evidence from RCT had been in snake bites since 1964, which is quite astonishing. And really, the pool of data from RCT comes from in snake bites from around 2,000 patients, which is nothing, is minuscule. And there's no global standard of care. And on top of all the challenges that there are with the efficacy, safety, and quality of the antivenom, it's a big challenge that needs to be addressed. So the any snake trials has been designed with a practical statistical design funded by the Wellcome Trust in the UK, and is being put together and developed, is set up at the moment to evaluate the safety and the efficacy of existing as well as new antivenoms in sub-Saharan Africa only, and is really there to develop a new evidence base for the policymakers. And again, like we did for NIHR and explained for NIHR, we're not looking at saying A is better than B, but in practice wanted to rank these antivenoms, looking as well at eliminating the worst ones. We're looking at different clinical syndromes, and we're looking also at incorporating a dose optimization within the practical design. And it is so important to have discussion with regulators early on. We're engaged with ARF. Some of you might be familiar. This is a WHO initiative to support the harmonization of approval of clinical trials in sub-Saharan Africa. And this is why we engage with them so early, hoping to start recruitment this time next year. Yeah, this is just a very simple trial design schema for the any snakes trial. As you can see, it's much simpler. There isn't a second line randomization as in NIHR. And really what I'd like to leave you with is there's a lot to learn, a lot more to do. I think just as a final couple of slides, there's been opportunity to talk about training. I wanted to say there's a number of courses that we organize and lead at the CTU on the conduct of platform trials. There is a whole MSc program in running clinical trials, as well as the stats. We have MAMS Clinic, which we offer to everyone to come and discuss the sort of challenges and opportunities with trials that you are perhaps designing and leading to conduct. And finally, because our work is a lot around LMIC and resource limited settings, we also created a capacity strengthening hub with a lot of resources free to access and with a lot of components about adaptive trials. We're looking for mentors as well. So if there's anyone who wants to join our LMIC mentoring scheme, we'll be delighted to take you on board. And thank you very much. Sorry for a round of time.